We'll continue with Chapter 3D. We're going to now do protein synthesis. So, protein synthesis is really the job of the cell. It's making proteins and all the things that proteins do for us as enzymes to drive reactions, as structural material, as metabolic material. And it's really important things. And what proteins are long sequences of DNA, long sequences of amino acids in a very specific order. Those amino acids basically are like letters of the alphabet, and a protein is like a word or even a sentence made up of those individual letters in a specific order. There are 20 amino acids, and we put them in a certain order. So getting them in the correct order is the job of the DNA. So one strand of DNA in a piece of chromatin will have the instructions for assembling many different proteins. Basically, a gene on that DNA is that segment of the DNA with the instructions for making one protein, one polypeptide. Now, there are 4 nucleotide bases on DNA. There is A and T and C and G, adenine, thymine, cytosine, and guanine. And that's not enough to code for all of the amino acids. So what we do is we group them in groups of 3 that we call triplets. And so each triplet codes for a specific amino acid. And so if there was 100,000 amino acids in a protein, then there will be 300,000 nucleotide bases in the instructions to make that 100,000 triplets. Now, we call the 2 parts of protein synthesis transcription and translation. The words are exactly what they sound like. When you transcribe something, you rewrite it in a different form. A medical transcriptionist will take a tape recording of the doctor's voice, of the radiologist's voice, and type it out. So she's not altering anything, it's just changing the form of it. It says the same thing. That is a transcription. Transcribing is copying things out, and usually in a different form. Transcribe, across, right. Translating is taking that information and doing something with it, changing, making sense of it, translating. Think of it. We'll go back to the cookbook analogy that I used. Each strand of DNA is a cookbook. We have 46 strands of DNA. So we have 2 copies of each cookbook in each cell. There are 23 cookbooks and each gene is a recipe for making one protein. If you've got precious cookbooks and you don't want to get them destroyed, you don't bring them into the kitchen. What you do is you open the cookbook to the right recipe, you copy it out, or you photocopy, or you somehow make a copy of that one recipe that you're going to cook. You close the cookbook up, you put it away where it's kept safe, and then you take the copy out into the kitchen, where you then cook the recipe. And you spill crap on it, and you rip the paper, and you basically abuse the paper in the translating of it. But it's okay, because all it is a temporary copy that you're going to use while you're in the kitchen. And it's no different with this. The DNA is the master copy, it's the master book, and it's kept safely behind the nuclear envelope, in the nucleus. What we do is we take the bit of the DNA that we are going to be using, and we copy it out onto something called messenger RNA. And the messenger RNA leaves the nucleus through the nuclear pores, and it goes out to the ribosome, where it is run through the ribosome, and the amino acids are assembled in the correct order. There's a couple of types of RNA. There's 3 types of RNA. The first one is called messenger RNA. And really, it carries the recipe for the single gene, for the single protein, from the DNA in the nucleus out to the ribosomes in the cytoplasm. That's why it's called messenger RNA. But the messenger RNA has to interact with something, and it has to interact with the nucleotides of RNA at the ribosome. So we call this ribosomal RNA. So this is a structural part of the ribosome that lines up and kind of holds the messenger RNA in place while it is being read. Now, there's also a type of RNA called transfer RNA. And what transfer RNA does is that it picks up free amino acids, and it is the code for that amino acid to line up with the corresponding codon on the messenger RNA. So a triplet on the DNA makes codons on messenger RNA, and then the anticodon of tRNA, of transfer RNA, bonds with the codon and brings the amino acid to the correct spot. So those are the 3 types of RNA. Translation doesn't happen without them. So, transcription is the making of messenger RNA. So the sequence of bases, of nucleotide bases on the DNA corresponds to a base sequence that's complementary, so it's the opposite on messenger RNA. So if the DNA has a triplet of TCG, then the messenger RNA codon that would be formed off of that would be, if I said TCG, they would be AGC. So the rest of this slide is talking about the biochemistry of how it happens. We're not so worried about it. I care more that you get the base sequence. So there's a polymerase that unwinds the DNA template, and all the nucleotides join together. And it ends up looking like this. So a piece of messenger RNA is made here. So if you look at this, so we can zoom in. Okay, you can see that the nucleotides for the RNA are starting to fill in on the DNA, and you end up with a complementary strand of messenger RNA with codons on it that are complementary. They're the opposite to the triplets on the DNA. Now there is one slight variation on this, and that is RNA. It doesn't use T. Instead, it uses a nucleotide base called uracil. Uracil is a lot like thymine, but and it still bonds with A, but the other end of it is slightly different. So because RNA has a different backbone, you see that it's purple here and it's blue there. So anytime that the codon has an A, the triplet has an A, the codon will have a U. Anytime that the triplet has a T, the codon will have an A. Anytime the triplet has a C, the codon will have a G. Anytime the triplet has a G, the codon will have a C. So the difference is that U is substituted for T's. Other than that, it's very similar to DNA replication, DNA synthesis. There we go. And all RNA is that way. So you end up with the enzyme going on to the DNA. So there's an initiation phase, then you start to unwind it, you start to make the messenger RNA as this enzyme runs, and you end up with a complete piece of messenger RNA with codons on it. So translation is converting the sequence of nucleic acids, so the codons on the messenger RNA, into a sequence of amino acids in the protein. So each triplet on the DNA, three-base sequence of the DNA, is represented by a codon on the messenger RNA. And each codon codes for a specific amino acid. So if the codon is UUC, it's phenylalanine. If the codon is UUG, it's leucine. If the codon is AUG, that means start, or if it's already started, it means methionine. If the codon is AAA, then it's lysine, etc. This chart kind of gives you a breakdown. There are 20 amino acids, but you can see that there are far more possible codons than that. So oftentimes 4 codons all code for the same amino acid. So the messenger RNA attaches onto the ribosome and then starts to move along the ribosome to that start codon. Then a large ribosomal unit attaches, the ribosome becomes functional. So the anticodon of the transfer RNA then binds to the codon that's complementary to it. So the anticodon, we go back to here, so the anticodon on the transfer RNA that and the transfer RNA is say it's carrying alanine here. So, and the codon is GCA. The piece of transfer RNA that's carrying the alanine will be CGU. So that the anticodon really is the same as the DNA triplet that originally coded, except there's U's instead of T's. That's really the difference. So the codon is the complementary to the triplet. It's also complementary to the anticodon. The transfer RNA really acts like a taxi that carries the amino acid to the site. And the order is dictated by the order of the codons on the messenger RNA, so that it ends up looking like this. So this is a piece of transfer RNA with the anticodon. This is the transfer RNA with the anticodon bound to the codon, bringing its amino acid in place. This is where it is picking up the amino acid. So the amino acid gets attached to the transfer RNA. The transfer RNA carries it to the ribosome. It plugs in, drops off its amino acid, leaves without an amino acid, goes back up here, and picks up another of the same amino acid. This is happening constantly. So all these transfer RNAs are either having just dropped off an amino acid, or an amino acid is in the process of being picked up. So the messenger RNA comes out the nuclear pore. The amino acid is attaching to the transfer RNA. The codon is in the ribosome. The next amino acid is being added to the chain of this growing polypeptide, which is going to be a protein. It gets read. So, the endoplasmic reticulum is really very much involved. So the messenger RNA is kind of moved through the endoplasmic reticulum. So it doesn't have to go all over the place. Remember I said the endoplasmic reticulum is really in contact with the nuclear pores. So therefore, the ER just keeps everything in line. The forming protein is part of this. Things can be added, sugar groups, fat groups, etc. Then the protein ends up going to the Golgi apparatus, where it's packaged up and shipped out. What's interesting is that these ribosomes are so close together that many of them are doing the same piece of messenger RNA, just at a different point in the sequence. This way we can be very efficient about how many proteins we're building. So the messenger RNA ends up finding the ribosome because of these signals. It's attached, the signal thing is released and gets recycled, it's used again, and you basically build the protein. So that's how proteins are made. Salient things are that in RNA it's U instead of T, that each amino acid on the DNA is coded in a triplet, 3 nucleotides together, A's, T's, C's, and G's are code for an amino acid. There's transcription happens, messenger RNA is made with the codons that are complementary to the triplets in that specific order. The messenger RNA is then brought outside of the nucleus through the nuclear pores to the endoplasmic reticulum, where it meets up with the ribosomes and is translated into that protein. Each amino acid is carried to the synthesis site, to the ribosome, by a piece of RNA called the transfer RNA. The transfer RNA has a series of 3 nucleotide bases that are complementary to the codon of the messenger RNA, and that is called the anticodon. The anticodon is the same as the triplet that originally coded for the thing, except U's and T's are different because the transfer RNA is RNA. The rest of this slideshow we're not going to bother with. We're not going to talk about aging or any of that stuff with it.